A very good morning to my dear students. I am Dr. H.S. Prithviraj. Today I'll be talking on a topic called transgenic plants. And this topic has been given in paper 8 of final year BSc. And this is one of the important topics in tissue culture part, and it's a part of biotechnology. So in this class, let us know what these transgenic plants are and how these plants are developed, what are its applications. So first let us know the definition of transgenic plants. So transgenic plants are referred as genetically modified plants in which genomic DNA has been modified. So we can modify the DNA of a plant to get a desired character or a trait in a plant. So this is also referred as genetically modified plant. So why we need to develop these transgenic plants? What are their applications in the field of agriculture? So the first reason being to improve the quality of the plant to produce a desired characteristics. So the second reason is to transfer genes from unrelated plant species, which is not possible traditionally, but it's possible and it's much faster and easier through biotechnological techniques. And this technology is useful for the isolation of the genes from many plant species. So these are some of the reasons that made us to develop the transgenic plants. Now, what are the steps in this transgenic technology? How to start with, how to go about? So we have to identify the useful gene. It's also called as GOI, gene of interest. So we need to locate the desirable genes in wild species or in unrelated plant species or in any other microorganism for that matter. So this is generally done in the research laboratories. So first step in developing transgenic plant is to identify a useful gene. Then we need to insert it into the recipient plant, or we can also call that as a donor. The gene of interest is isolated from the donor source and cloned in the laboratory. So this is again a laboratory technique that is used, right? So the next step in developing transgenic plant is to insert the gene into a target plant. So the cloned genes or the multiple copies of the gene of our interest are inserted into a host plant or the recipient plant. So there are two methods generally that are used for the insertion. It can be agrobacterium mediated gene transfer or gene gun bombardment. So this is again a laboratory technique. And another important step in developing transgenic plant is to identify the transgenic cells. Once these genes are inserted, we need to identify the transformed cell by selectable markers. And this regenerated plant is compared with a parent variety, and this should look like a parent variety. So next we need to transfer them to small scale field trials. So the transgenic plants that are evaluated for their performance, then these plants are generally tested in small fields. So the seeds of transgenic plants like cotton, soybean, these are generally tested in the laboratories, and these tests are also conducted with animals. And then after the small scale field trials, we have to grow them in large field trials. These transgenic plants are then multiplied in larger quantities, in larger fields, and superior genotypes are then released for their performance. So this chart speaks about the characters that have been improved in certain crop species. If you look into the chart, you will come to know the crops, their characters that has been improved, and you can find these crops showing resistance in the insect. They show resistance to diseases, they show resistance to herbicides, they have high quality, and they also show some antibiotic resistance to some abiotics, they show resistance to abiotic stresses. Now the crops being cotton, maize, tobacco, tomato, which show resistance against insects like Helicoverpa, Arbizera, some budworms, from fruit borers. So these genetically modified crops, they show insect resistance. And crops like tobacco, potato and tomato, they show resistance to diseases like brown spots, seedling blight, potato leaf rot, tomato mosaic virus and seedling blights. And some crops like cotton, soybean, wheat, potato, tomato, they also show resistance to herbicides. And you can increase the quality of these plants, particularly in potato, tobacco. You can increase the protein content, the starch content, etc. And some crops like tomato and tobacco, they also show abiotic stresses. Now this is another chart showing some of the achievements in the transgenic technology, particularly the cotton, maize, wheat, tobacco, potato and tomato, soybeans, rapeseeds, linseeds, alfalfa and petunia. They show some improvement over the normal plants. So these are the genes that have been transferred from various organisms. Now coming to the applications. So far you have understood what a transgenic plant is and what are the reasons for its development and how these are developed. Now another important concept of transgenic plant will be the applications. So why this has to be developed in agriculture, particularly in agricultural field, this has a major role in the form of its nutritional quality. So we can increase the nutritional quality of the crops by these transgenic plants. So these plants are, as I already told you, are resistant to insects, diseases, pesticides, etc. And they are also resistant to salty areas. They are salt tolerant plants, and they are also very useful in biopharmaceuticals. So the modified plants are also used to produce medicines and proteins. Now, coming to a few important examples where this has been done, I have already told you some of the crops have been modified genetically, and taking one of the examples like golden rice. So the golden rice has been developed mainly to enhance the keratin and iron content. So this has been developed by transferring genes, two genes particularly, from daffodil and one from the bacterial species. So golden rice, it is benefiting millions of people in the world, and those who suffer from health problems caused by vitamin A deficiency. So now we can see how this golden rice is different from the normal rice. You can see the golden color of the rice in this picture. So you can see how this golden rice is different from the normal rice. So now another important plant that has been developed is potato, where the protein content has been enhanced. So potato has shown more protein content in it. So this transgenic potato contains about 33% of the higher protein than the non-transgenic potato. So in that way we can increase the protein content of a crop. So this is also referred as protein rich potato. So you can see the protein rich potato. So the percentage of protein is more here than the normal potato plant. Now another crop that has been developed with the transgenic technique is tobacco. So nicotine free tobacco has been developed. So genetically engineered nicotine free tobacco does not synthesize nicotine in their leaf. It will lead to introduction of nicotine free cigarettes. So we can make cigarettes free of nicotine. In that way, this smoking will not cause any harm in the smokers, and it is one of the best applications in the field of biotechnology. So here is how the field of tobacco looks, and these are some of the aspects of transgenic plants. So I have taken only a small percentage of this technique because it's a vast subject. So I have taken a minimum time to cover up certain aspects of transgenic plants. I hope this video will help you in terms of your examination. So from the examination point of view, so these are certain aspects of this topic